the editor-in-chief of the journal MAPS.
As Victor mentioned, I'm Janice Reichert, Executive Director of the Antibody Society and the moderator of the panel. First, I'd like to welcome our panelists, which include all of today's speakers, as well as Ajay Royyuru, who is Chief Science Officer for Healthcare and Life Sciences Research at IBM. And we also have Andy Nixon, Vice President, Biotherapeutics Molecule Discovery at Boehringer Ingelheim. Welcome, everyone, and thank you for participating. As a reminder to attendees, the bios for all of our panelists are available via the event webpage, so please do have a look at that. And you may add questions yourself for the panelists to the Q&A box. So let's get started with the discussion. Panelists, the first question is, what recent milestones or advances in bioinformatics-driven drug discovery do you think are particularly important for the field? Anyone can dive in.
I'm going to say the obvious answer to give everyone else time to think of everything else. So we have to start by saying AlphaFold, because we would be foolish and silly to ignore the fact that that has not only in giving lots of structures of, all of the genomes, but also the architectures it has put out and what it has helped us to understand about what you can do with these methods is also a powerful part of that. It's not just it's done structure prediction. I don't think that's a finished question yet, but it's really helped us. But it's also helped us understand the types of methods, not only for structure prediction, but lots of things for dealing with the data. And the other thing that I will throw into the mix, which I think both Lucy and Konrad mentioned, is this evolution of the concept of the latent space of a thing. So the latent space of antibody sequences being a really nice example of that. And now everyone else has had a chance to think, I would be glad.
And I would add as well, Charlotte, that paratope prediction is really kind of moving us forward to be able to do in silico antibody discovery, which is something that I have wanted to do for the last 10 years. So I'm really excited with kind of where we are with, both AlphaFold as well as, this ability now to do paratope prediction. I think that's really going to move us forward.
And this is Ajay. I'm first very glad that the organizers have included me in the conversation. I learned a lot today, so this is wonderful to catch up. To answer Jan's question or add to what has already been said, I think this notion of actually creating a latent space representation that captures properties that you then want to sample from is a very beautiful development that is occurring in the field. And a progress that we all can learn from is actually how small molecule drug generation has already begun to occur quite successfully in that space. So I would say we learn from that to say, how can we actually bring that to a biologic generation? But of course, the complexity of properties that you have to learn or represent in the latent space is much richer, right? Or it is different, at least, and I would hypothesize that it is also much richer. So there is some learning that we have to go through. But there is success that has been achieved in small molecule generation from latent space representation that tells us that this is actually a useful direction.
Cool. Maybe if I just can add one thing. So I think that especially with regard to latent spaces, one thing that we are very advantageous with in antibody space is that NGS just can create quite a lot of data. Yeah, because there is quite a finite amount of proteins that are there in theory, but antibodies we can just keep sampling them. Yeah, so to just feed the models even more data, this can it seems that we can just keep going towards this 10 to 16, 20, whichever an estimate you like. What we would like to see if actually you're reaching an asymptote and actually sampling this space. Because I actually don't think that 10 to astronomical exponent is actually how many antibodies are there.
Great, thanks for those responses. Now I'll ask you about the biggest bottlenecks or challenges in silico drug discovery that are current. What right now is stopping or is a bottleneck or a challenge in silico drug discovery?
I mean, I wanted to pick up on the point that Charlotte made at the end of her talk in response to one of the questions. You know, one of the things that we are certainly doing is exactly what you described. So, you know, I'm global head of molecule discovery for biologics at BI. And we are modeling kind of outputs from our libraries, as well as you know, whatever that library is. And, you know, library could be from a phage display, or it could be from, you know, the output from an animal immunization. And, you know, for me, that is still a real bottleneck in terms of, you know, being able to model those sequences, generate structures, and do that in a kind of rapid and meaningful way. There's still kind of lots of work that you know, I think we can do there. So that for me is kind of a key challenge. The other thing as well is you know, and it's been mentioned several times throughout the symposium, is really that kind of high quality curated data. And you know, I mean, there's lots of things that we can be doing, and that I can certainly think of, and lots of things that were described here. But access to those high quality data sets is a real challenge. And you know, even for us in pharmaceutical companies, with people like me who are kind of beating the drum, it's still really hard to get those well curated data sets.
Bottlenecks, challenges from anybody else? Because the next question, go ahead, Charlotte, go ahead.
Well, I was going to say, I think I agree with everything Andrew's just said, which I don't think is that surprising. I suspect we all agree around this. I think there are also still challenges, and I think I should put this one there. I don't think it's a bottleneck, but I think it is still a challenge, that the field has not totally embraced the concept that computation is not just an additive. You know, it's not just I'll interfere here, or this code will help me make a prediction, or if I sit here and think about it, but it's like, it should be the thing that helps you think about changing the entire pipeline. So you don't, for me, I see this as quite a large challenge. It's not take my bits of ideas and just feed them in where they'll help you a little bit. Please do that. But actually, if you really want to change and make you know, a big step change in how good we are at this, it involves redesigning the whole process and pipeline, thinking about where we start from, not thinking about it in the way that we currently do, thinking about where the machines can actually help and build on that. So for me, that is also a kind of challenge to try and get people to understand and embrace that, because it's a big change for everybody.
With that, the next obvious question is, moving forward, in what direction should the field go in order to address the challenges or to advance the field? Like, how do pharmaceutical companies reorder their thinking to put in silico work at the front end of it, so that they are doing that first?
Well, maybe I can say, like, first of all, like, and they are using, like Charlotte said, like in the bits and blobs to like, you know, a more and more bigger extent. But yeah, it's I cannot say, because, like, you know, I do not work in those companies. Like, you know, this is very, very, very different from actually changing the entire process. Yeah, so using the different bits and blobs helps increasingly, but it needs a bit of like, you know, a revolution to actually, like, you know, change, you know, the approach culture, and, like, you know, where the capital goes, and, you know, like how the entire calculation of how you make a new drug is made, you know, like even like absolutely like science, how you do it aside.
So Andy, that seems like it's a perfect spot for you to hop in.
It does. You know, it is really a mind shift change. I mean, this is kind of, I think, the topic. You know, there are a lot of dinosaurs like me kind of still roaming around the kind of pharmaceutical industry who, you know, grew up with immunizations or phage display, and so this approach of in silico or computational antibody discovery is really new, and there's an enormous amount of resistance in organizations to these kinds of approaches. But I think it is the future. And for me, the question that I'm trying to answer for people is why? Why do we need to do this? Why is this gonna be better for us? And really, for me, it's about opening up the kind of druggable target space. Being able to kind of access things that have otherwise been really challenging. There are lots of projects that I could think of where we know from knockout studies that a particular antigen is involved in the disease pathology, but we have a hell of a time making the protein, and so really struggle when it comes to immunizations or kind of the kind of more traditional antibody discovery approaches. And so I think that for me, opening up the druggable target space using computational approaches, I think is really attractive. And I think that answering that question of why do we want to do this is, I think a key lever in changing the mindset. But I think, changing the mindset is really the kind of cultural task that I think, people like myself have in our organizations.
Very well said, Andy. I would add a little bit to the how. I think the traditional thinking, particularly in such very, very hard problems of antibody discovery, have been approached as a straightforward experimental protocol of explore, select, refine, and then move ahead with it, right? And computational methods are actually, they lend better to parallelism, to risk mitigation at different stages of the process, not necessarily in this linear way. So a concept that I think the experimental and computational researchers have to get familiar with, comfortable with, is how do you do co-design? You actually have to progress with all tools available and not just pit one against the other. You can't, I like it that the rigor that we are establishing right now is, can you predict a structure that is as good as a crystal structure? Okay, that's rigorous, but it is not going to be how experimentalists will consume models in future. They will just use the model rather than do a crystallography attempt, right? If the model is good enough, then you basically are doing some kind of, use the right thing for the right decision and move on. Okay, so you're trading off half an angstrom of resolution, perhaps, or maybe you're trading off on some variable region flexibility. But if that's not what matters to you, don't bother with trying to do a crystal structure. Just use what you can get your hands on, right? So that's kind of a core design notion of how that we have to get comfortable with. But indeed, I think the field is not there yet. The field is not there with regard to accepting those trade-offs because we haven't established that rigor. It's beginning to happen. I hope it'll happen in the next few years, but we are not there yet.
I would just add that, what I'm seeing is, obviously it's going to take some practical successes to really start to change people's minds, particularly doing things that are very hard to do. I mean, I was recently at a conference where somebody was presenting design of an antibody that binds two different antigens in the same antigen binding pocket, a very difficult task. And then afterward, when you heard people talking, it's sort of like, is this possible? Like, is this, an incredibly difficult task for which it's not obvious how you do that experimentally, select that molecule. I feel like that the mindset, there is some change happening. It's slow and it's going to take very practical examples of success to show, in my mind anyway. And also, it's not clear that computation is essential for every task, right? So the more proper application of computation to solve kind of the right problems that are just like Andy just shared, a target that's very hard to produce, a cross-reactive antibody that hits an epitope that's very difficult to target, using experimental methods. It's just like very hard to do. And there's many, many examples of those, right? So I think we also need to be thinking about what we're doing, right? And where are those wins going to be that are going to continue to change people's minds?
I think I was going to say something very similar to some of that, which is that thing about one of the best ways to change people's minds is to have a moment like an AlphaFold 2 moment, and we haven't had one of those yet, where there's a demonstration which is so clear and so simple to understand that people can, even if the future isn't here, you know, let's be clear, AlphaFold 2 does not accurately predict every protein structure. You can see the future. And we as an entire community have not had that moment, I think, in this space yet, but I think it is on its way. I was going to add something kind of, I mean, a little bit added to this about bottlenecks that relates to the culture change, which is around that data piece. One of the reasons the culture change is so important is at the moment, all data is generated, and that's not fair, I should not say all, most data is generated as decided by a human for a particular thing that they are interested in based on what they thought was a good idea. And that's basically how the process works. If we want, like Konrad mentioned, to build a better antibody language model, you wouldn't randomly sample space. If you wanted to build a better structure predictor, you wouldn't randomly sample space, and you certainly wouldn't sample it very precisely around one structure and think about that loads. You'd start thinking about all the places you didn't know that might not be useful as therapeutics right now, but if I did that, then my model will become loads better and more powerful. So the change in mindset is both that kind of process thing, but also even around how we generate data, because all data is not created equal. So for me, a bottleneck on data is not just make me more and keep it clean. It is, how do I generate data that is actually useful? Because, as you know, all data is not equal in terms of how much it will balance.
Yeah, and just to sort of follow that up for a second, I remember when I talked about the AAV study, when we proposed generating or synthesizing sequences that had been sampled at random, our experimental collaborators who were going to do the experiment thought we were completely crazy. I mean, they did it. They were like, we're going to do it because, we sort of talked it through and everything. And they were like, okay, we'll do it. But they really thought that this was like a waste of money and effort and time. And actually, in hindsight, that data set has been remarkably useful. And so this was a suggestion that came, purely from sort of a machine learning. It wasn't my suggestion, purely from kind of machine learning, computational perspective. And it was kind of nuts, and it was 90% dead, that data set. And it still sort of surprises me that it was useful, but it was really useful. And I think that sort of speaks a little bit to the kind of decisions that Charlotte is talking about, that we have to think carefully, sort of, and we don't necessarily anticipate having to sort of think about them, right? I mean, I don't know. Yeah, that. I guess just thinking about the sort of AlphaFold 2 moment, to what extent does there need to be like a CASP for there to be an AlphaFold 2? I don't know what do people think? Like,
I think that's basically like a solving Capri for antibodies, would be a moment like that, pretty much. Or even like just, calling whether it binds or doesn't bind. Yeah, like how are you define bind? Yeah.
But I mean, does Capri sort of fulfill that role? Like, I mean, is it a sort of, is it established enough? Is it famous? Like,
I think you don't necessarily have to have a competition to do it, but what you do have to have is a really simple question. The reason that AlphaFold 2 was such big news is not just because it's cool. There are loads of other cool things that have been done. It's because everyone can understand. It's really easy to understand what's been done. And so for me, bind, not bind is quite a nice question, even though we would have all sorts of delicacies around that, a bit like we have about whether you compare to a crystal structure or not. But fundamentally, if somebody said bind, not bind, and we could do that, everyone would understand. So I think if you're thinking about a big moment like that, what you're thinking of is, what is a question that it's so easy to grasp the question that when you can do it, it's really obvious to everybody it has been done. Because most of our questions, it's not obvious when we've done it. I can't, Konrad said, we can't really tell how accurate we expect H3 modeling to be. How accurate do I expect paratope prediction to be? Because there's going to be some flex in that as well. How, if I got one, I'd be slightly worried, if you sort of mean.
Great. Thanks very much for those responses. Moving on to a question about industry and academia living together. What specific areas in biopharmaceutical informatics are there where industry and academia can complement each other or collaborate? And I'd also like to draw in a question from an attendee along similar lines. What do any of you wish was different about teaching the curricula in various majors to better train people for drug development that's heavily computational? Are there key skills you think should be taught at the undergraduate or graduate level? And that seems like a perfect spot for industry to help inform what's going on in academia. Is there any chance of that?
So maybe I'm going to say that, like, as a person that actually went through the path of being in academia and actually being trained together with the industry. So this actually, the being able to have the academic freedom, this is extremely good. But actually being able to be exposed, this was my doctoral program with Charlotte, to all the industry and actually the practical questions, that those academic questions are being put to. This was extremely good, because academic freedom, does not actually translate to the freedom of asking the same questions in the industry.
So, I think an aspect of beyond the individual to the community of practitioners in the industry and practitioners in the academia, I wish that we could together achieve some higher degree of defined interfaces for modularity of functions. And this is a nascent area of research, right? We're seeing all the proliferation of tools and data aggregations, et cetera, which is indicating that there is actually a growing area of work happening. But how do we mature it to a point where defined interfaces allow for very rigorous tool assessment and, more importantly, actually interplay between the tools to occur? That's very important for industrialization of the research workflow, because today what happens is a practitioner, and I'm speaking, as a practitioner in the past, a practitioner actually has to understand the nuances of every individual tool that they either buy or in-source from some open source collection, et cetera, and kind of have to figure out how to string it together, when to use it, when not to use it. And that's an inhibitor to the maturation process. It is very good for curiosity. It is very good for pinpointed investigations. But if you want to say that, I want to speed up my overall end-to-end, then, all these actually become inhibitors because each one is a point of specialization. So none of us can alone solve that problem. But as a community, of course, we can tackle that. If we were to be more defined in posing the module of problem that this is addressing, and what are the accepted interfaces to either pose the problem or assess the answer to that problem. And I'm not expecting that all that maturity is going to come in just one day. But if you begin to move in that direction, the maturity that will occur for the consumption of these research tools in the industry would be more of plug and play. Okay, and then you can see that the acceleration that is going to occur in the consumption of these technologies would be much faster, because you're not trying to assess every tool by itself. You're actually assessing it in the consumption, you're assessing it in an end-to-end manner. And there are some accepted ways in which you can plug and play with them. So that's kind of a workflow orchestration that has begun to occur in many other areas of data-driven science applied to industries. And I believe that can come to this sort of biologically based problems as well.
I'm going to answer that with my academic hat on and go, to do that, sitting on the other side of the fence for a moment, would be the necessity of the pharmaceutical companies from their side releasing their data in a format so that you could have that kind of assessment happening. Because there's a real limitation about, I could easily win at lots of the problems that can currently be posed with public data because it's easy to cheat. And by cheat, I just mean you overtrain or you don't separate very well. But if I separate very well, I start to have very little data, and it's hard for you to tell which method is very good until you actually take it in-house. I would kind of like to move this on slightly into the general stuff as well, though. So I have two hats. I am an academic at Oxford University, but I am also CSO at Exscientia. And I always think that the interface between academia and industry is much easier than most people think of it as being. I mean, a huge percentage of students in my group are sponsored by loads of different companies. I found that companies really like working with academia. It's not some kind of, oh, go away kind of, they're really keen, that they are happy to talk and share as much as they are able with all of these things. And sometimes all I think needs to happen is for all of us just to be happy in that space and not worry that it is always slightly confusing about exactly what can and can't be done and how this will work out. But when people ask me about how we can complement each other and collaborate, the obvious things are things like we ask different types of questions. We have different end games on this. You know, my end game is to publish a paper. That's probably not the end game usage inside the pharmaceutical company. But we have the same aim overall, which is to have the best possible method for solving a given problem. And that just requires us to be able to understand that problem properly from both sides. So I always think with this, the answer is talk to people, share your information as freely as you are able. That's very easy speaking as an academic. I can share it very freely indeed. But doing that allows us to have really good conversations about what's important and then work out where the expertise comes from to answer these. Because there isn't kind of particular expertise in academia that isn't in industry, and there isn't, or vice versa. There are just pockets of places where you can find information and work with each other.
Thanks very much for the responses. Question specifically for Lucy and Ajay. What role can big tech play in helping biopharmaceuticals companies incorporate biopharmaceutical informatics into their processes with the aim of accelerating drug discovery and development and making it more efficient?
Yeah, so I'm more than happy to sort of open this and then yield the floor to Ajay. So in the time I've sort of spent at Google, we've actually collaborated quite a bit with sort of various pharmaceutical companies, and our goal there really has been to sort of establish that these approaches can actually be useful. And I think, you know, there's not really sort of, you know, it's not the case, I think, that like Google has any aspirations in sort of the pharmaceutical direction, at least not any that I know anything about. It's obviously a big company, but you know, for us to actually establish that these approaches can be useful, we really have to work with the people, you know, sort of the experts actually using them, and we have to work on real problems. It's too easy to kind of sit, you know, in a sort of slightly machine learning community and sort of solve problems that we think are important, but that don't actually kind of reach across into practice and translate into what people actually need. And you know, we certainly don't have any of the experimental expertise that would be required to sort of demonstrate that these approaches work. And we want to really work on kind of real problems and show we can do this. And so I think there is a role to be played there in sort of doing sort of proof of principle work and showing that things can be done. But then I also think at some point it probably has to transfer, you know, that we kind of don't, I don't think Google wants to keep doing that forever, to sort of provide an ML consulting service in any sense. And I doubt that's a comfortable place for pharma. You know, it seems that they're very, very, very careful about data and you know, sharing that kind of stuff with big tech seems scary. So I think there needs to be some kind of transfer of knowledge and expertise, and sort of I see that happening to some extent, but I think it was mentioned earlier by somebody that they're kind of, you know, or maybe with one of the questions, there needs to be kind of a different approach from the start, maybe, in terms of, you know, you can sort of bolt things on, but at some point you have to kind of really change things, and I think that's harder to achieve. I don't know, Ajay, what do you think?
Thank you, Lucy. Yeah, so first I would say I concur with what Charlotte and you are saying, that industry researchers and academia researchers, they're both researchers. They're both looking at the science problem. They're just bringing a different set of, perhaps a different set of tools to tackle it on occasions. But sometimes the tools are not very different either, right? So it's not like they're speaking a completely different language or coming at it with something that is unknown. So there is commonality, which obviously today's session actually shows there is so much of that commonality. what industry can bring additionally, at least speaking from IBM and IBM research point of view, is industry is accustomed to thinking, the tech industry is accustomed to thinking about scale first. They are not looking to say, you know, I have a small spanner and I'm just going to turn one little nut with it, right? Because if that is all that they end up doing, then it just doesn't make a difference anywhere else. So industry, the tech industry is accustomed to actually picking problems that if you were to solve would have a scaled impact of some kind. That's kind of a fundamental assumption that pretty much everybody who chases problems in tech industry makes. So that's a strength actually for this field, right? Where if you pose the right kind of problems that have inherent scale, like today we saw the advantages you get by actually bringing scale, not just in volume of data, but in diversity of data, as an example. Or, you know, I heard today very beautiful instances or examples where performance of computing is not fast enough. You have a solution, but it doesn't perform fast enough. Okay, so the industry, tech industry is actually accustomed to tackling such problems. So making something 10x faster or 100x faster is something that the tech industry attempts to do all the time, right? The very first time you implement an algorithm or you go and do something new, you know, it does not achieve the performance that you want it to, but you don't give up. You actually go wring out every little bit. You do caching, you do all sorts of representation of information to go wring out the very last bit of performance so that you can get all that it. So that's a strength that tech industry typically always has. And we now need to actually invite. So that's why, you know, I return back to this notion of modularity of problem space. And how do you zoom in on specific bits of the problem that actually lend themselves to that kind of scaled, industrialized, heavy lift? And I think each of those are achievable. You know, my vision, as I shared previously, is you ought to think of the workflow, not just of a single unit of functionality. You ought to think of the workflow and piece by piece actually achieve that kind of acceleration. So, you know, we in IBM Research have begun to think of this as accelerated discovery. That's our vision, right? So technology actually achieves an acceleration of the discovery process itself. And it's not one technology, it's not one silver bullet, but you got to have all these pieces coming together in this manner. And it's not going to be one company alone, right? It'll take all of us to do that together. So that's actually, we feel that is the ingredient, the essential ingredient to achieve the transformation that we began this panel discussion with right? Where we are all aiming for a transformation. And that transformation is not going to happen by just nibbling at one thing at a time. We really have to ask, what are all things that can be pursued and then in combination actually achieve that transformation?
Thanks very much. All right, so final question real quick in the last minutes. And maybe the answer already is this transformation you're talking about is, can you pick quick? What excites you the most about future applications of biopharmaceutical informatics?
If I can say, it's basically right now it's becoming realistic that, you know, it is going to become a driving force here, because for some of my students, like in bioinformatics, like, you know, I actually show a Forbes, like, you know, cover from like the 80s, yeah, like in computer design drugs, yeah, didn't happen, yeah, but right now it's essentially like with all the data and tools currently being there, short of the, you know, the culture change that like it was mentioned, I think that like, and this is within grasp. Not us, but I'm pretty sure there's someone.
Yeah, I mean, it's, for me, this is a really exciting space, you know, and it's really starting to come to maturity. And like I said, I mean, I've been kind of wanting to do in silico antibody discovery for over 10 years now, and it feels now like it's within grasp. And so to me, you know, I would expect in the next 5 years that we are really able to do it. And that for me is just tremendously exciting.
I'll offer a big picture view that, you know, some of us who are trained in traditional bench biology, you know, have through the last decade, certainly, or actually 2 decades perhaps, been made to realize that biology actually is an information science. And it is actually nothing but information science. We use experimental means to just get to that information, but essentially it is information science. We are observing through experiments, we are hypothesizing through human intelligence at the moment. And so if we were to, what excites me is if we were to flip this into, it's an information science after all. So can we hypothesize? And I like one of the comments made in a talk about not hypothesizing once, but actually hypothesizing at scale. I think it was Charlotte, you were talking about making 1 million hypotheses. Not all million are going to be equally good, right? But that is what we have to get better at. If I had the ability to throw 1 million hypotheses, how would I say which ones should you trust, right? That is where we need to go to, because then the scale of technology becomes our asset.
Well, thanks everyone for awesome responses. So in closing the panel discussion, I would like to thank the panelists for all of these informative responses to the questions that we posed. As a quick reminder, in addition to the presentations, the panel discussion was recorded and it will be made available on demand soon. So I'll now turn the show over to Victor, who has the closing remarks.
Yes, and I would just like to say thank you again to the panel members, and so to everyone joining, the speakers, the panel members, and also the attendees, of course. And thank you also to the Antibody Society and the planning team for hosting this event and for marketing it across all the platforms. And yeah, and as was also just said, the recordings will be made available, and the next TAP symposium will be on November 1st, and that will focus on harnessing cytokines for cancer immunotherapy. And I think with that, we can close. Thanks, everyone. Have a good evening or rest of the day. Bye-bye.
Thank you.